were there different side effects observed with that agent compared to larotrectinib that you know of?
I think definitely there was, as I recall, there were definitely different side effects. I think less hepatotoxicity, but other side effects such as diarrhea, et cetera, were observed that we didn't necessarily see as frequently in Laro.
So,
And likely that's probably from the inhibition of ROS and so forth.
Okay. So, and as far as sort of safety, the safety signals then, other than just the side effects?
Yeah, I think the safety signals are very similar to Laro in the sense that overall Entrectinib also is a very safe drug. Mm-hmm. And the studies, I think, have been going longer on with Entrectinib than in the Entrect population than the Stark studies. But I think the overall safety signals are very similar. Both of these drugs are not very toxic.
Right.
And so.
And I appreciate your comment that you really can't compare because the trials are very different,
yeah.
and you really can't compare directly across them. And hopefully as we get more data, we'll actually be able to know a little bit more. Did the ROS-ALK inhibition from Entrectinib affect the efficacy in lung cancer? Because we often see those obviously those mutations in lung cancer, but you're not going to see both TRK obviously and ROS.
Correct. It's an effective drug in ALK-ROS patients, and ROS specifically. Mm-hmm. I think they've reported response rates up to 60% or higher. I think...
is it more active in ROS mutated patients than in TRK, for instance?
The data would suggest that it's more effective in ROS, or at least the waterfall plots and response rates.
Right.
I'll be honest with you, I don't know the answer to that
question. And I think that the data for the TRK alone is so few that it's really going to be hard to say. But I guess we'll know again when the data sets get bigger.
Yeah.
Would you have a certain population that you might pick Entrectinib versus larotrectinib, or are there a certain patient population because of this data that you would say, if they have CNS mutation, would you pick one over the other?
Yeah. Again, I'm biased. I would, if you have an Entrect fusion, I would go on larotrectinib.
Okay.
If you have a ROS, if you have ALK-ROS, I mean that's debatable, right? Because there's a lot of osimertinib. Sorry, not osimertinib. Other drugs right now. Osimertinib is EGFR inhibitor. Right. There are other drugs right now in this class that are incredibly effective. You'd have to ask the lung experts really to figure out where does that fall in which space, right?
Yeah.
And
so we'll only have to split off that activity and study it completely separately, I think.
In terms of how I expect to use this class of agents, my experience to date is exactly one patient who was enrolled on a clinical trial of Entrectinib and actually did in fact have a response, sharing that patient with another colleague, so I'm not primarily taking care of this individual. But the presumption is if we identify this marker at the get-go as part of next generation sequencing in their initial workup, more likely than not we will treat this individual with the targeted agent. We'll defer other forms of systemic therapy. That's not true of all molecular abnormalities. For instance, CMET mutations or amplification preferentially use chemo first or chemo combined with PD-1 or PD-L1 inhibitors. But in this case, if Entrect rearrangement has been identified, these individuals will get larotrectinib, and presumably once it's approved, Entrectinib. And at that point, we'll start asking the question, can we sequence these agents? What's the activity of one after the other? Currently, those are essentially unknown. Ultimately, though, one expects that these individuals will become TKI refractory, and in that group, chemotherapy necessarily remains the final common pathway. Whether to give chemotherapy with angiogenesis inhibitors like bevacizumab, whether to continue the TKI along with the chemotherapy. Again, a number of open, unanswered questions exist.